Hi, I'm Sam Ozminkowski, and this is Identifying Microbial Drivers in Biological Phenotypes with the Bayesian Network Regression Model, work done with Claudia Solis-Limus. We know that microbial communities are among the main driving forces of biogeochemical processes in the biosphere, and we would like to understand how the composition of microbial communities affects complex biological processes in soil, plants, and humans alike. In order to do so, we present a Bayesian network regression model originally developed by Guha and Rodriguez on brain connectome data. Unlike brain data, however, in which every brain region appears in every sample, microbiome data is often very sparse. Most microbes are not present in most samples. We performed a thorough investigation of whether this model is suitable for microbial datasets on a wide array of biologically realistic simulated data, and implemented the model as an easy-to-use Julia package so that biologists can use it on their own data. This model treats the response as the product of an adjacency matrix describing the relationships between the microbes and a coefficient matrix describing the effect of each relationship on the response, plus some random error. This model allows us to estimate these edge coefficients as well as the probability that each microbe itself is influential on the response. The simulation study presented here is described in the graphic on the top right of the poster. We begin by simulating a 30-taxon phylogenetic tree and a true coefficient matrix. The phylogenetic tree is simulated using the R package APE. For the coefficient matrix, we first determine if each microbe is influential on the response by drawing from a Bernoulli distribution with either 0.3 or 0.8 as the probability that each node is influential. Then, we set each edge coefficient as follows. For every pair of influential microbes, we draw from a normal distribution with mean 0.8 or 1.6 and variance 1. For edges where one or both microbes are not influential, we set the edge coefficient to 0. Using two different values for the mean of the distribution allows us to test how the results of the model differ when the magnitude of the effect each edge has on the response differs. For each of 100 or 500 samples, we randomly select 8, 15, or 22 microbes from the phylogenetic tree. We generate the adjacency matrix for each sample using the inverse of the phylogenetic distance between each pair of microbes in the sample. This way, microbes which are more closely related to each other have higher values in the adjacency matrix. If a microbe is not in the sample, any cells in the adjacency matrix corresponding to that microbe have a value of 0. Finally, we calculate each response as the Frobenius inner product of the adjacency matrix and the true coefficient matrix, plus some random error, drawn from a standard normal distribution. Some brief results are presented here. Figure 1, in the bottom left corner of the poster, shows 95% credible intervals for influential edges in two cases, both with strong edge effects and dense adjacency matrices. The top plot gives results for the denser coefficient matrix, with the bottom giving results for the sparser coefficient matrix. For both plots, the left panels show the truly non-influential edges, while the right panels show the truly influential edges. Edges found to be influential by the model, defined as having 95% credible intervals that do not include 0, are shown in dark blue. As you can see, in this case the model gives no false positives and few false negatives. Figure 2 shows the probability of influence for each node, for 8 and 22 microbes and 500 samples. Truly influential nodes are in dark blue. Again, we see the model does quite well here. Using 0.5 as a naive cutoff, there are no false negatives or false positives, except in the case of sparse adjacency matrices, a sparse coefficient matrix, and weak edge effects, in which case there are 2 false negatives. Additionally, the truly influential nodes tend to have probabilities of influence that well exceed 0.5, and the non-influential nodes tend to have probabilities of influence which are far lower than 0.5. The Julia package is available for use now and can be found by scanning the QR code in the bottom right corner of this poster. Further results, including those from a second simulation study, are presented in my talk of the same name, so please view that for additional information. Finally, I'd like to thank Claudia as well as the other members of her lab. In addition, I'd like to thank Doug Bates and Rick Lankow for their help on this project, and of course, thank you to the Department of Energy and the USDA for supporting this work.